Hello, my name is Adam Myers, and this is my lab report on the isolation of Itsy Bitsy spider bacteriophage from Chesapeake Bay oysters for the purpose of genome sequencing. A bit of context for starters. Vibrio harvii is a bacteria which is notable for infecting different types of marine life, predominantly fish and other types of invertebrates. It can cause a whole myriad of different diseases within these organisms. And a vibriophage is a virus which seeks out and destroys bacteria of the Vibrionaceae family, including Vibrio harvii. It is because of this attribute that makes scientists believe that vibriophages might be a valuable asset in fighting diseases caused by these Vibrionaceae bacteria as a non-chemical sort of antibiotic. And in this lab, we're going to attempt to isolate the DNA of a vibriophage for the purpose of eventually sequencing it and creating a indefinitely long-lasting sample of said DNA. The methods of how we're going to do this. For starters, to cultivate our bacteria, we used two methods. We had streak plates on plates of agar to maintain a solid sample of our Vibrio harvii bacteria, as well as liquid cultures, which we made using test tubes of marine broth. To isolate our bacteriophage, we did this through essentially taking the biological material out of an oyster and filtering the material um, out of the oyster to create a liquid which we then mixed with Vibrio harvii and a marine agar to create an agar plate in the hopes of forming plaques which we could harvest phage out of. And we did this with two different oysters, one from a place called Blue Point and one from the Chesapeake Bay. Of these, the Chesapeake Bay oyster seemed to have the most viable results, and thus it is what we extracted the Itsy Bitsy spider phage from. Now, to purify that, dilutions were made of said phage in a buffer, and these were also made into agar plates multiple times over to get a pure sample of our phage. Once we had done this, we were able to create a lysate, which is essentially just a liquid stock of our phage for use in future experiments, including a spot titer, which we used to estimate the amount of phage per milliliter of our lysate, which we did through creating a spot titer plate of agar of different dilutions. Then, also using our lysate, we were able to view our phage under the electron microscope. We were able to visualize it as well as take photographs of said phage, which we will see in a minute. Additionally, with the same lysate, we were able to extract DNA out of the phage. And from here, we were able to use a gel electrophoresis device to get a readout of the length of our phage's genome, as well as make a negative 80 degrees Celsius sample of our DNA, which can be stored indefinitely. Now, the results of our experiments. So, in terms of phage isolation, as previously mentioned, we were unsuccessful in yielding plaques of phage from the Blue Point oyster. However, we were successful in harvesting phage from the Chesapeake Bay oyster, as seen in this image. These plaques are what we harvested the Itsy Bitsy spider phage from. Now, for purification, we were able to yield plaques from most of the attempts of purification, usually ranging within the 10 to the negative 2 or 10 to the negative 3 dilutions of phage buffer. However, on some attempts of purification, there were difficulties in obtaining plaques at all on any of the plates, and so several of the purification phases had to be entirely redone. So we were successful in creating a lysate of the phage, and using this lysate and our spot titer plate, as seen here, we were able to determine that there were 10 to the 8th plaque-forming units per milliliters of lysate. The results of our electron microscopy. So as you can see on the right here, we were able to visualize the phage and get a sense for its size using the electron microscope and our sample of lysate. And then we were successful in extracting DNA out of the Itsy Bitsy spider phage. And so we were able to use the gel electrophoresis device and compare our phage's DNA to a DNA standard. And through doing this, we were able to assume that the Itsy Bitsy spider phage has a genome length of around 10,000 base pairs long using our key and comparing it to the DNA standard as seen here. And then that faint little mark right there is what was left by the Itsy Bitsy spider phage. And as previously stated, using a decoder of sorts, we were able to deduce that the Itsy Bitsy spider phage's genome is around 10,000 base pairs long. Some concluding points to wrap up. The goal of creating a sample of the phage DNA was successful. We were successful in extracting the phage, and we were successful in isolating it. However, there were, of course, complications throughout the process, most notably in purification, in which multiple stages had to be entirely redone. However, these challenges were overcome. They may largely have been human error, although it was unsure and still is unsure what exactly caused them. As for future implications of the Itsy Bitsy spider phage, it is possible that with additional research, it could be used in potential future phage therapy options, as previously stated, for different marine life in fighting Vibrio harveyi-caused diseases. And that could be beneficial not only to our ecosphere in terms of having more healthy environments here on Earth, but it could also be helpful economically in helping to preserve fish and other invertebrates which may be used as seafood or have other uses to